So as we have discussed, the genetic code's main function is to control the production of proteins in an organism's cell. And so a gene is a DNA molecule that contains the information to code for one specific protein. Your DNA may have many different genes on it, but each individual gene may contain several hundred to millions of nitrogen bases. Remember, nitrogen bases are adenine, thymine, guanine, and cytosine. The order of the nitrogen bases matters in the process of producing proteins. So groups of 3 DNA bases code for one specific amino acid. And there are 21 different types of amino acids. This is how proteins are made. So cells use information from a gene on a chromosome to produce one specific protein. All of this takes place in the ribosome, which is in the cytoplasm. So the question is, how does the information on the chromosome get out of the nucleus into the cytoplasm? Because DNA lives inside the nucleus, and this process takes place outside of the nucleus in the cytoplasm. Well, there's a special kind of copy of the DNA that is called RNA, and it is a genetic messenger. So it's a little bit different than DNA. It's only one strand instead of 2 that are wound together, and it has something called uracil instead of thymine. So again, we still have A, C, G, but instead of T, there's a U. But just like T and A pair with each other, U and A would pair with each other in this case. There are different types of RNA. There is messenger RNA, which are copies of the coded message from the DNA that is made in the nucleus, and it exits through the pores in the nuclear envelope, and it carries the message to the ribosome in the cytoplasm. And then there's something called transfer RNA, which actually works to carry the amino acids to the ribosome and help add them to the growing protein. We will watch a video about this process, and in the video, the things that kind of look like green butterflies are the transfer RNA carrying the amino acids, and the things that are yellow strands are the messenger RNA. So the very first step is to translate the code. What has to happen is the DNA has to unzip so that the base pairs are exposed. One strand of the DNA directs the production of the mRNA by pairing up with the DNA bases, which is shown here. So you can see that there is an unpairing occurring right here. The unpairing occurs right here. You can see this is the DNA, and then you can see that the messenger RNA is formed by matching up with the unzipped portion of the DNA. Just like a puzzle piece, the nitrogen bases match, as we've discussed before. The mRNA is then able to leave the nucleus, sorry about that, and it's able to then enter the cytoplasm. So the DNA never leaves the nucleus, but information, individual genes that need to be expressed in that specific cell are copied into mRNA, and it is that mRNA that leaves the nucleus. We've talked about the DNA being like a library, and if you're studying ancient Rome, you don't need to check out all of the books about how to cook or about other countries, but you do need to check out the books about Rome. Just like that, for each individual cell, only the genes that are needed in that particular cell are the ones that are copied into mRNA, which then leaves the nucleus and travels to the cytoplasm. The next step is that The mRNA actually needs to attach to a ribosome, which is in the cytoplasm, because it is what provides the code for the protein molecule to be formed. So here's a picture of the ribosome. The purple thing is the ribosome, and here is the messenger RNA. What you can see is that groups of 3 nitrogen bases are paired with, are basically red, like a ticker tape. And you can see here that there are these little green figures. Those are representing the transfer RNA. So inside of the cytoplasm, there are these things called amino acids. There are 21 of them. Each individual transfer RNA codes for one specific amino acid. There are 3 nitrogen bases here that code for that. So it goes around into the cytoplasm and it carries that amino acid to the ribosome. And then, when the right combination on the messenger RNA is read through the ribosome, it's paired up with the proper transfer RNA. Next, the tRNA attaches to the mRNA and reads the message. So it's pairing the 3-letter codes to the bases and creating a chain of amino acids. So here you can see, as this ticker tape is being read through, you've got the transfer RNA or tRNA matching, and then it's delivering the amino acid. That's the beginning of a protein. A protein is built by these amino acids. So as this one gets attached here, this transfer RNA, just as that one, will leave and will go find the same amino acid again and come back. And if that same chain that it matches up with of nitrogen bases is needed again, it would come in and deliver the amino acid to start building the protein chain. The protein molecule grows longer and longer as each tRNA puts the amino acid it's carrying into the protein chain. So you can see here that the chain of amino acids group together and they create a specific protein. Once an amino acid is added to a protein chain, the tRNA is released into the cytoplasm and it can pick up another amino acid. It picks up the same kind of amino acid always because it is coding for a specific group of 3 nitrogen bases. It is important to note that mistakes can happen along the way, and those mistakes can be called mutations. So a mutation is any change in a gene or chromosome that causes the cell to produce an incorrect protein during protein synthesis. Mutations can occur at different times. They can occur in body cells, and those are not passed on to offspring. But if a mutation occurs in a sex cell, meaning a sperm or an egg cell, those can be passed on to offspring. And there are 3 different kinds of mutations that can occur. A substitution is one in which one base pair is substituted for another base pair. A deletion can occur when one base pair accidentally doesn't get copied and it's just deleted. Or an addition can be when one base pair is added. So there's actually an additional base pair. And these mutations can occur in the process of the mRNA being copied. So for example, if we were to look here at this one, you can see that the correct order is supposed to be CCCTTT. But here, what we have is a substitution. Accidentally, in the copying process, this A combination was substituted when it was supposed to be a C. And what this does is it changes the type of amino acid that is pulled to be added to the protein chain, which creates an incorrect protein.